Welcome to today's Explainer. We've got an absolutely fascinating journey ahead of us. We're looking at how we take these literal microscopic lab miracles, specifically with stem cells and neural repair, and try to translate them into a grand, almost sci-fi vision of reprogramming the way we age. If you're into the bleeding edge of regenerative medicine, you are absolutely in the right place. Let's get right into it.
Our main blueprint for this deep dive comes straight from a really comprehensive presentation on translational neurotherapies. We're going to dissect this whole complex process of translating iPSC-derived treatments from the lab to the real world. We'll look at the actual clinical outcomes, the super frustrating bottlenecks, and some truly novel frameworks researchers are proposing right now. Basically, we're looking at three distinct phases of this bench-to-bedside translation. First, the proven empirical evidence, the hard data showing this science actually absolutely works. Then, the harsh manufacturing realities, why making these therapies viable at scale is so incredibly hard. And finally, the systemic bedside paradigms, the big picture strategies for the treatments of the future.
So, here's our quick roadmap for today. Number 1, the bench. Number 2, the bottleneck. And number 3, the bedside. Okay, let's jump in.
Starting off with part 1, the bench and the empirical evidence. So we're kicking things off with the actual breakthrough cellular mechanisms that prove this tech does what it claims. And it all starts with a very strict engineering target. You can't just throw a bunch of random stem cells at a spinal injury and hope for the best. You start with pluripotency, those iPSCs, and you force them down a very specific lineage. The target here is to create neural progenitor cells, or NPCs. Now, for these to actually be effective as a therapy, you need them to be extremely pure, over 80 to 90% purity. And not only that, you need a highly specific functional ratio, like 1 neural cell to every 10 glial cells, just to properly control the physiological symptoms once they're inside the body. It is highly, highly engineered.
The actual experimental setup runs on a really strict timeline. Phase 1 is a 30-day in vitro differentiation process right in the lab, where they're constantly testing for action potentials. Then comes Phase 2, a 5-week in vivo engraftment phase. They take these validated NPCs and inject them locally into surgically induced spinal cord lesions in SID mice. Those are mice with severe immunodeficiency, which is super important because it ensures the human cells aren't just instantly rejected by the host's immune system.
Actually, really quick, just to make sure we're all on exactly the same page before we hit the results, iPSCs stand for induced pluripotent stem cells. What we're doing is taking regular, mature adult cells, completely reprogramming them back into a blank, embryonic-like state, and then basically telling them to grow up all over again into brand new neurons. It's literally biological alchemy, and it totally sets the stage for the recovery data we're about to see.
And wow, the in vivo outcomes are striking. We're talking about a statistically significant recovery in actual motor function. The mice treated with these NPCs showed highly quantifiable improvements in reflexes and grip strength when you compare them to the untreated controls. And the cellular engraftment data completely backs this up. When they use immunofluorescence tracking at that 5-week mark, it confirms robust cell survival right there at the local lesion site. The cells aren't just surviving, they're actively living and integrating.
But how exactly are they doing this? Well, mechanism 1 is all about restoring the electrical architecture, because these transplanted cells aren't just sitting there taking up space. They are electrophysiologically active. The data confirms they're actually generating mature action potentials, meaning they're acting as fully functional neurons capable of transmitting signals. And crucially, they also trigger localized remyelination. Basically, they're repairing the biological insulation on the neural wires, which is absolutely vital for getting that motor recovery we just talked about.
Then we have mechanism 2, which honestly is perhaps even more fascinating. It's microenvironment modulation. Using single-cell RNA sequencing, researchers can see that these cells act as crucial bridges for the neural circuitry. Think of it kind of like a biological seesaw. The transplanted NPCs actively use cellular signaling, like the Wnt pathway, to alter the local immune environment. They actively suppress hostile pro-inflammatory markers, and at the same time they boost anti-inflammatory ones, like ARG1. They essentially pacify the whole injury site, creating this permissive, calm zone where neural regeneration can actually happen.
All right, moving on to part 2, the bottleneck, dealing with the manufacturing realities. So the science we just covered is spectacular, right? But it begs a really obvious, absolutely crucial question. If we can basically cure spinal cord lesions in mice with such high efficacy, why isn't this therapy in hospitals right now? Why the massive gap between a lab discovery and actual commercial availability for human patients?
Well, the answer lies in what we call the translation bottleneck. This really breaks down the friction we're facing. First off, time and cost. Culturing these cells takes 12 to 30 continuous days in a highly controlled lab. And those extended timelines mean exponentially higher costs and way higher risks of contamination. Second, impurity clearance. Remember that 80+% purity requirement? Eradicating all the leftover undifferentiated cells before human trials is a massive engineering hurdle. And third, stage instability. These cells are incredibly sensitive. I mean, the slightest tiny change in their environment can cause them to spontaneously mutate or turn into the completely wrong type of cell.
To really wrap our heads around this instability, we have to look at a pretty profound concept. As noted in the source material, in biology, measurement alters the subject. It's kind of like quantum physics, honestly. You literally cannot verify a living cell's current state without fundamentally altering the exact environment it lives in.
So this brings us to the core issue, the quantum measurement problem of cell quality control. To commercialize this, to get it to hospitals, you have to scale up to millions of cells, and that requires rigorous quality control. But here is the massive dilemma. The exact moment you extract a sample from a flask to verify its specific markers, you strip it away from its precise, delicate, perfectly balanced fluid environment. And doing that causes an identity shift. It is exceptionally difficult to stage lock these cells, meaning to freeze them in that exact neural progenitor state while you test them without them degrading or differentiating into something else entirely. It's just an immense manufacturing paradox right now.
Which brings us to part 3, the bedside, looking at systemic paradigms.
For our final act here, we're elevating the discussion. We're moving from localized spinal repair to the massive vision of whole-body rejuvenation. But to understand systemic therapies, we first have to contrast two very different anatomical spaces. First, you have the intravascular space, your bloodstream. This is highly accessible. Clinicians can easily deliver therapies here just using a standard IV. But then you have the interstitial space, the microenvironment deep inside your specific organs. This space is largely inaccessible without highly invasive surgery. And this contrast, this is the fundamental challenge of whole-body repair.
The current strategy for systemic rejuvenation relies entirely on crossing this boundary, what's called the intravascular interstitial axis. Because direct intervention deep inside the organs is practically impossible, the goal is to alter that accessible intravascular compartment, maybe using factors from endothelial or mesenchymal stem cells, to indirectly influence and reprogram the totally inaccessible interstitial organ spaces right across the barrier. However, we run straight into a brutal mathematical hurdle, the gradient of efficacy. See, stem cells secrete these really powerful soluble repair factors. If you extract these factors into a concentrated fluid in the lab, you totally bypass the risk of injecting whole cells into a person. But mathematically, you might have a massive concentration of, say, 10 to the negative 9 in your lab flask. Once you inject that systemically into the human bloodstream, it vastly dilutes before it ever reaches the target organ. The concentration might drop all the way to 10 to the negative 12. So how on earth do you achieve a therapeutic dose when you're fighting that kind of massive systemic dilution?
Well, the answer involves fundamentally rethinking how cells actually heal. We have to overcome cellular dormancy. As our tissues age or take damage, our cells get trapped in this dormant state known as G0 arrest. Now, the traditional thinking of just giving a continuous, steady biological dose to fix them, it turns out that's highly inefficient. Instead, the whole objective has to shift. We don't need a steady drip of medicine. We need a massive, initial, highly potent biological stimulus just to break that arrest and violently wake the cells back up.
Which leads us to a truly thrilling concept, the spark hypothesis. Think of a car engine. You don't need to hold the key in the ignition the entire time you're driving down the highway. You just need a single massive spark to start the engine, and then it runs completely autonomously. Biology is looking really similar here. We don't need continuous dosing. A massive, intermittent trigger, a biological spark, can knock those aged cells right out of G0 rest. And once they're activated, the body's own autonomous healing cascade can just take over and do the rest.
So let's synthesize our journey today. First, the foundational science is undeniably validated. We know iPSC neural progenitors can engraft, they can restore myelination, and they drive real, measurable functional recovery. Second, the reality of manufacturing these things is going to require immense innovation. Overcoming the biological observer effect and figuring out how to stage lock cells are the true hurdles we face right now. And third, the future is incredibly systemic. We are moving beyond local injections to trigger autonomous whole-body rejuvenation using that incredible spark hypothesis. As we wrap up today's explainer, I really want to leave you with a provocative thought to ponder. Based on all the mechanisms we've just mapped out, from neural electrical repair all the way to triggering dormant cells back to life, are we merely finding better ways to treat localized injuries, or are we actually on the verge of reprogramming age itself? Thank you so much for diving deep with me today. Keep exploring and keep learning.